When an organism reproduces, eggs and sperm are formed through a basic biological process called meiosis. Meiosis is kind of a cell division that results in four haploid cells from a single diploid cell, and the number of chromosomes are halved. And during this process, it's important that the number of chromosomes are maintained uniquely to a specific species over a generation. Meiosis is divided into two consecutive phases, meiosis 1 and meiosis 2. In meiosis 1, the separation of homologous chromosomes, basically one parental and one maternal chromosome, occurs. These chromosomes are firmly paired to form tetrads, and when the homologous chromosomes cross over, a transfer of genetic material occurs along with boosting of genetic variety. So when we first get into meiosis 1, we have prophase and metaphase. That's where basically they start basically going through their thing. So in metaphase, triads are aligned in the metaphase plate, and this happens randomly, independently, just based on assortment. We then move to anaphase 1, and this basically guarantees every daughter cell will receive a mixture of parental and maternal chromosomes, since homologous chromosomes are drawn to the opposite chromosome, or pole, if you would say. In telophase, the cells are then divided into two haploid cells, with each one having a set of chromosomes. After the telophase, there is a cytinase phase, which we're not going to talk about. So we'll just move into meiosis 2. Meiosis 2, the sister of the chromosome, or another word for it is chromatid, is parted, and this similarly occurs in mitosis. We then move to prophase. In this prophase, the chromosomes are condensed once more, then as similar as into the metaphase and anaphase. Chromosomes are aligned in the metaphase, and then the chromatids in anaphase are then attracted to the opposing pole as in anaphase 1. Same thing occurs in anaphase 2. In the typhlophase, we once again will have two haploids divided into four haploid gametes. And then we have this final stage here. Meiosis is really brought into light or better illustrated when we consider Mendel's experiment with pea plants. And basically, this experiment was focused on the bloom color, and they found that the purple bloom color was dominant over the white. And we see how meiosis contributes to this, genetic inheritance. In Mendel's experiments with the pea plants, we discover the law of the law of segregation. During meiosis, a trait alleles is separated, and this then results in the distribution of one ally among each gamete. And then every cell in a diploid organism contains two alli, one from each parent. The gametes bearing either the dominant P, which is the purple, or the recessive P, which is the white. And then this in turn produces a plant with a genotype. Basically, it has both recessive and dominant. The homologous chromosomes end up carrying one dominant P, and the other ends up carrying a recessive P. And then they're divided into distinct cells in the meiosis first anaphase. When it comes to the total dominance in Mendel's P plant experiment, the expression of the ally, which is the lower P or white, is really hidden by the existence of the dominant P or ally, which was purple. And as a result, only genes with two recessive P's were displayed white. All others that had a dominant P or a lowercase p ended up being purple. If they had a two dominant P's, they will end up being purple. The only time the plants will end up being white, even if they have a mixed match of recessive and dominant, is if both of them had recessive alleles. In conclusion, in order to produce haploid gametes and maintain a genetic variety, meiosis is necessary in processing these genes, and the independent assortment is important as well. Um, the gametes mixing in during fertilization gives offsprings their diploid status back, and then the distribution of allies in these gametes allows whatever the offspring inherits to have a specific pattern of genetic material or features that is independent but also dependent on the parent.